In this episode of Toxicity Tests, we're going to look at pyrogenicity testing. This is a definition of endotoxins from the Alltox website. Endotoxins are one of the main pyrogens society needs to test for. However, other pyrogens do also exist, and of course they need to be tested for as well. It reads, endotoxins are fragments of gram-negative bacterial cell walls that can produce fever and chills, and possibly a fatal reaction when introduced into the circulation of humans and animals. Since even sterilized products can contain residual bacterial fragments, each batch of injectable drugs and some medical devices must be tested for the presence of bacterial endotoxins. This testing is called pyrogenicity testing. This is a statement from the European Commission's Director General Joint Research Centre. It's from the Institute for Health and Consumer Protection, and the statement is on the validity of in vitro pyrogen tests. It reads, at its 24th meeting held on the 20th to 21st of March 2006 at the European Centre for the Validation of Alternative Methods, the Scientific Advisory Committee unanimously endorsed the following statement. Following a review of scientific reports and peer-reviewed publications on the following range of in vitro pyrogen tests, you can see them there from 1 to 5, it is concluded that these tests have been scientifically validated for the detection of pyrogenicity mediated, that's caused by, gram-negative endotoxins, and quantification of this pyrogen in materials that are currently evaluated and characterized by the rabbit pyrogen tests. This slide shows the second part of the statement, which reads, these methods have the potential to satisfy regulatory requirements for the detection and quantification of these pyrogens in these materials subject to product-specific validation. The test methods have the capacity of detecting pyrogenicity produced by a wider range of pyrogens, but the evidence compiled for and considered within this peer review and validation process is not sufficient to state that full scientific validation of this wider domain of applicability has been demonstrated and confirmed. Thus, the above test methods can currently be considered as full replacements for the evaluation of materials or products where the objective is to identify and evaluate pyrogenicity produced by gram-negative endotoxins, but not for other pyrogens. This endorsement takes account of the dossiers prepared for peer review, the views of independent experts who evaluated the dossiers against defined validation criteria, supplementary submissions made by the management team, and the considered view of the peer review panel appointed to oversee the process. And it's signed by Thomas Hartung, Head of Unit, European Centre for Validation Alternative Methods, Institute for Health and Consumer Protection, Joint Research Centre, European Commission, ISPRA, which is in Italy, and it's dated the 21st of March 2006. This slide is from the Canadian Council on Animal Care. In the top corner you can see the title Conventional Test Method, and it is the rabbit pyrogen test. The description of the alternative test, well, one of them here is the human whole blood in vitro pyrogen test, and the validation status, as we have just heard, is that it was validated by the European Centre for the Validation of Alternative Methods for the detection of pyrogenicity caused by gram-negative endotoxins in 2006. If you look under US, the US found the test suitable by the Interagency Coordinating Centre for the Validation of Alternative Methods for use to detect gram-negative endotoxin in human parenteral, which means drugs that you inject into the body rather than taking them orally, drugs on a case-by-case basis, subject to validation for each specific product to demonstrate equivalence to the RPT, which is the rabbit pyrogen test. So the conditions in the US for this test to be used are much harder than they are in the EU, where it's been pretty much seen as a direct replacement to the rabbit pyrogen test for endotoxins. Under regulatory status, there's no information from either the European Union or the United States. So it's just another indication that it's one thing to validate a test, it's another thing for it to be accepted by the regulatory authorities. Anyway, the next column says effect or potential effect on animal use, and it says replacement when used to test for gram-negative endotoxins. This slide shows pretty much what the other slides showed, except it gives a description of the test. Remember, there's 5 of these tests, but I'm just going to look at this one test here. It says the test is based on the principle that white blood cell monocytes differentiate into macrophages and release inflammatory cytokines when exposed to bacterial endotoxin. The test exposes whole human blood to the test substance and measures macrophage cytokine release using an immunoassay. So that's the principle of the test. Unfortunately, we're not going to have enough time to look at the other 4 tests. We're now going to look at how these tests were received in the United States. This is an ICCVAM test method evaluation report from May 2008. The ICCVAM, I think I got it a bit wrong earlier, I said centre instead of committee, so it's the Interagency Coordinating Committee on the Validation of Alternative Methods. So I thought I'd quickly clear that up. Anyway, it says the SACATM, they are the Scientific Advisory Committee on Alternative Methods, and this is their evaluation of the in vitro pyrogen test methods that we looked at earlier. This slide shows the final minutes of the SACATM meeting convened on June the 12th, 2007, and it's quickly worth reading through this. It says overview of the ICCVAM evaluation of in vitro pyrogen test methods. Dr. Richard McFarland, US Food and Drug Administration, Pyrogenicity Working Group Chair, presented an update on ICCVAM's ongoing evaluation of 5 in vitro human cell-based pyrogen test methods nominated for review by the European Centre for the Validation of Alternative Methods. Pyrogenicity is defined as an increase in body temperature following the release of pro-inflammatory cytokines, e.g. interleukin and tumour necrosis factor by leukocytes. Anyway, it goes on. Pyrogens may be found in processing and packaging materials, chemicals, or parenteral pharmaceuticals, biologicals, and medical devices. Bacterial endotoxin, a component of the outer cell wall of gram-negative bacteria, is one of the most potent pyrogenetic materials. Pyrogen testing is important to prevent the introduction of endotoxin or non-endotoxin pyrogen-contaminated products into humans or animals. The last paragraph reads, currently there are 2 accepted pyrogen tests. The rabbit pyrogen test, which measures a temperature rise in rabbits injected with a test substance, This test can detect both endotoxin and non-endotoxin pyrogens. The bacterial endotoxin test, also referred to as the Limulus amebocyte lysate test, detects endotoxin by its ability to activate a serine protease catalytic cascade. We'll look at that test a little bit later on, but unfortunately it's not a total non-animal test because you have to get the blood from a horseshoe crab. This slide is still looking at the comments from the Scientific Advisory Committee on Alternative Methods. It reads, Dr. Thomas Hartung from the European Centre for the Validation of Alternative Methods joined the public for this specific agenda item because of a conflict of interest as a patent holder for the methods. 3 of the in vitro test methods were based on his research, and he had coordinated the validation study prior to joining the European Centre for the Validation of Alternative Methods. So you can see here that there is a slight conflict of interest. I mean, he's trying to get these tests to be validated by the Americans, but he also has a patent. He is also a patent holder. Anyway, it goes on. It says Dr. Thomas Hartung was pleased that the European Pharmacopoeia, which is a big book with all the drugs that are allowed in Europe and how drugs have to be tested and the weights and the measures and everything you have to know about the drugs. There is a US Pharmacopoeia as well. But anyway, getting back, he was pleased that the European Pharmacopoeia will hold a peer review panel to review and accept these methods. He was disappointed with the outcome of the peer review panel meeting. He noted that pyrogenicity tests are very expensive and the approval and release of a single product can cost several $100,000. The validation studies were set up to assess whether the new tests would outperform the old tests within a set threshold. I'm not entirely sure why Dr. Thomas Hartung was disappointed with the outcome of the peer review panel meeting. It was probably to do with how the validation studies were to be performed. Anyway, it goes on. Only 50% of the samples would be positive in the most sensitive rabbit strain. All of the in vitro assays have an accuracy of around 90%. He outlined 6 points where the BRD had been criticised. The BRD stands for the background review document. So here we have a scientist saying that the rabbit test, even the most sensitive rabbit strain tests, are only 50% accurate, whereas the in vitro assays, the 5 in vitro assays that Dr. Hartung has brought over from Europe to America so they can ascertain whether they would be useful themselves, he says that they have a 90% accuracy rate. So, you know, this comparing it again to the rabbit test, which is only 50% accurate, seems a futile endeavour to me. Anyway, let's have a look, see what the next slide says. This slide is still taken from the Scientific Advisory Committee on Alternative Methods comments on the 5 in vitro tests. It reads, in conclusion, the proposed test methods for which data sets have been provided perform better than the BET, which is the biological, what does that stand for? Sorry, bacterial endotoxin test, which we will look at later, and the RPT test, which is the rabbit test, which we've been looking at mostly. It goes on, Dr. Hartung proposed that the rabbit assay be replaced with the in vitro assays because the rabbit test cannot match their performance as reported in the background review document. The background review document is probably the document that was handed to the Americans from the European Centre for the Validation of Alternative Methods. which is largely what they're looking at here. So they're looking at the evidence basically that's come across from Europe. In this slide we get the thoughts of Dr. Burrill, who is a leading discussant on the Scientific Advisory Committee on Alternative Methods in America. What Dr. Burrill says is, there was no question about the usefulness of pyrogenicity testing and the urgency and importance of validating these tests. In combination, some of these tests will contribute to the reduction of animal usage. One major deficiency of present pyrogenicity testing is that the RPT, the rabbit test, only detects about 50% of the endotoxins, which, let's face it, is quite a major deficiency if you think about it. This is the last slide from the comments from the Scientific Advisory Committee on Alternative Methods, which obviously is the American response to the in vitro tests which have been brought over from Europe. Dr. Brown says she does not believe that it is necessary to run in vivo assays in parallel with the in vitro assays. She is unsure how one can run a regression analysis with one test that is 90% accurate and a second that is 50% accurate. She questioned whether it is necessary to validate an in vitro test against an animal test that is not as accurate as the in vitro assay itself. Scientific anti-vivisectionists have been pointing this out for a long time. The in vivo assays in parallel with the in vitro assays, they're asking to test the substances on rabbits in parallel with the in vitro assays. Now this is a real problem because the animal studies, the animal tests have never been validated for their accuracy, so the poor people, the poor scientists around the world who have invested a lot of time in producing these in vitro assays are then asked to compare them with something that has not been validated and is not accurate. So how can they ever win? You know, it's a completely ridiculous state of affairs. This slide from the Canadian Council on Animal Care, in the column where it says Limulus amoebocyte lysate test, aka bacterial endotoxin test, that is in the alternative test column. On the blank column on the left hand side, that's where the rabbit test would be. So this is an alternative to the rabbit test, but is also usually used at the same time, it seems to me. It's like a double test that they use on pyrogenicity tests. You can see in the EU it's accepted by the EU and listed in the ECVAM, which stands for the European Directorate for the Quality of Medicines and Healthcare / European Pharmacopoeia. In the US it's accepted by the FDA and listed in the United States Pharmacopoeia, and in Japan it's the same. So this is an alternative to the rabbit test, but it doesn't really fulfill all the credentials that we would prefer in the animal rights community, because it means taking the blood from a horseshoe crab. Anyway, in the replacement column it says relative replacement, replacing more sentient animals with animals that current scientific evidence indicates have a lower potential for pain perception. So it's basically saying the crab has a lower potential for pain perception than the rabbit, and therefore it is a better replacement test. Anyway, let's have a look at the test itself. There you go, at the top you can see it says conventional test method, rabbit pyrogen test. Below that it says alternative test method, which is the one that we've been talking about, the Limulus amoebocyte lysate test, aka bacterial endotoxin test. It says this test uses blood from horseshoe crabs, which is very sensitive to bacterial endotoxin and coagulates in its presence. The Limulus amoebocyte lysate is incubated with test substance and in some cases other substrates which aid in endotoxin detection or quantification. It continues, several recognized techniques exist including gel clock methods, chromogenic methods and turbidimetric methods. And at the bottom, as I said before, the replacement, it says relative replacement, replacing more sentient animals with animals that current scientific evidence indicates have a lower potential for pain perception. So let's have a look, see how the horseshoe crab fares when it's having blood taken from it, which is needed for this test. On this website about the horseshoe crab, the first paragraph reads, how does the horseshoe crab protect the public health? And I'm going to put another slide up so we can read that a little bit easier. It says, the horseshoe crab plays a vital, if little known, role in the life of anyone who has received an injectable medication. An extract of the horseshoe crab's blood is used by the pharmaceutical and medical device industries to ensure that their products, e.g. intravenous drugs, vaccines and medical devices, are free of bacterial contamination. No other test works as easily or reliably for this purpose. This slide, taken from the same website, says, the associates of Cape Cod and other LAL manufacturers have studied horseshoe crab mortality following the bleeding procedure and have found it to be quite low, less than 3% when compared to controls handled similarly but not bled. So that means that 3% of the crabs die because they are having undergone this procedure, which is 3 in 100, which is, you know, it's not that great, is it really? Anyway, less than 3% when compared to control. There are no records of a horseshoe crab dying during the bleeding process itself. Other studies conducted by government agencies and universities indicate a mortality of 10 to 15%. However, the horseshoe crabs in these studies were not handled as carefully as those collected by the LAL industry. I'm going to end this episode on pyrogenicity by looking at a response/comment to the independent peer review panel in America, which was set up to appraise the efficacy of these 5 in vitro pyrogen tests. The response is from Dr. Thomas Montag Lessing and Dr. Ingo Spritzer of the Paul Illich Institute in Germany. These 2 doctors were specifically concerned that all the available data on pyrogenicity testing hadn't been taken into consideration by the American Independent Peer Review Panel. Before we go on to these concerns, I'll just read out the bottom part of this slide. Dear Dr. Stokes, please find below our comments to the Independent Peer Review Panel report. 5 in vitro test methods proposal for assessing potential pyrogenicity of pharmaceuticals and other products. In this slide, which is going to be the last slide, Dr. Thomas Montag Lessing and Dr. Ingo Spritzer are complaining about a lack of data. What they say is that they want some reference data, which hasn't been forthcoming. It reads the PRP, which is the peer review panel, stated a summary of reference data demonstrating whether substances were shown to be pyrogenic in humans either passed or failed the rabbit test or the horseshoe crab test, or the in vitro test would have been useful. Absolutely crucial, I would have said. There are data published regarding adverse reactions in patients caused by drugs which were negative in the rabbit test. That's a false negative in the rabbit test, so the rabbit test didn't show it up, as well as in the bet test, which is the crab shoe test, Again, that's a false negative, but they go on, but could be tested positive using in vitro pyrogen test. That's a true positive. So this data does exist. Where was it? I think that's what they're asking for. They then go on to give a couple of examples. The first one, they say the first event happened with a tick-borne encephalitis vaccine, which induced fever up to cramps and hospitalisation in around 50% of the recipients. As mentioned above, both the rabbit test and the crab horseshoe test remain negative, false negative, in testing the product. In contrast, this product produced positive, false positive, true positive, results with blood samples of 50% of the donors applying the in vitro pyrogen test. And he names that test, the one we looked at actually, the whole blood test. The second example concerns a dialysis solution which caused aseptic peritonitis in the patients. Again, both the rabbit test and the bet test, which is the horseshoe crab test, were negative, whereas the in vitro pyrogen test could identify the incriminated batches. So it's proving itself to be more accurate here, and you have to start asking yourselves, would you want to be taking these things after they were tested on rabbits with their incredible 50% detective rate, or would you prefer to be taking the in vitro tests with their 90% detection rate? And I feel that Dr. Thomas Montag Lessing and Dr. Ingo Spreitzer have summed it up really well in the last sentence of this slide. It reads, these two examples demonstrate that the in vitro pyrogen tests are in certain cases superior to the RPT, i.e. the rabbit test, since they are working in the homologous, I'm sure that's not how you pronounce it, system, homologous, yeah, maybe it is, i.e. human indicator cells and fever pro-inflammatory reactions in humans. That is, they're using human cells to predict what is going to happen in humans. I can't stress how important that is for accurate results.